Hello YouTube! Today we're going to look at this idea of a shadow biosphere, which has been proposed most prominently by Carol Cleland and Paul Davies. Now, one of the basic assumptions that we find in modern biology is that all life on the Earth shares the same basic biochemistry and descends from a common origin. All organisms are biologically related. The shadow biosphere is the proposal that there might be as yet undiscovered life on Earth that uses a radically different biochemistry and that perhaps arose completely independently from life as we know it. So the question is, could this be the case? What are the prospects for a shadow biosphere? Now the first question here is, well, what exactly is life? If we want to determine whether or not there could be life that is radically different from the life we know of, then we need to have some idea about what the distinction between life and non-life is. Otherwise, how will we know if we did encounter radically different life? The question of what exactly is life has generated a great deal of debate in philosophy of biology, and it would require a whole other video to go through all of the proposals. But I think we can state at least some important properties that we're looking for. First of all, the feature that most biologists take to define life is that all living organisms are composed of cells. So this is cytoplasm enclosed in a membrane, and the cytoplasm contains the biological machinery for replicating itself. This cellular definition of life is why many biologists say that viruses are not alive, because viruses are not cellular. They can't replicate themselves, they have to hijack the cells of other entities. Now, I think we might worry whether this cellular definition is too narrow, but I think that what we do require is at least something like a cell, something closed off from external perturbations. We need a system that's capable of maintaining a distinction from the wider environmental processes. Now, maybe that wouldn't be a cell, strictly speaking, but you do need some sort of cell-like thing. Another crucial aspect of life is metabolism. Metabolism involves two processes. First, there is catabolism, which is the breaking down of organic matter. This is what allows the organism to get at the raw materials of the environment. Second, there's anabolism, which is the use of these raw materials to build up new molecules. Metabolism is basically just the set of chemical transformations by which an organism obtains energy and other essential molecules. It's very difficult to see how you could have any kind of life without some sort of metabolism. Third, a fundamental feature of life is reproduction. Of course, not all organisms in fact have the ability to reproduce, but I think we can say that all living organisms should have been brought about by the process of reproduction. Now, reproduction is going to require some means of information storage. In life as we know it, this is achieved using DNA and sometimes RNA, but in principle, life could use other machinery. So anyway, this gives us a broad idea of what life is, of what we're looking for. If we're looking for a shadow biosphere, we need a system that is protected in some way from external perturbations in the environment, that exhibits metabolism, and that is produced by reproduction. So, the next question, what could alternative life be like? In what ways could a living organism differ from life as we know it? Now, there are all kinds of options here. I won't try to go through every single one because it's an indefinite list, but let's explore a few possibilities. First, amino acids. Amino acids are the building blocks of proteins. Proteins have a very wide range of uses for organisms, so amino acids are central to life. There are over 500 known amino acids, only 20 of which are commonly used in life on Earth. Now, not just any sequence of amino acids will produce a functional protein. Many, perhaps most, amino acids could not build functional proteins. Nevertheless, it's probably the case that more than 20 are viable. Actually, there are a couple of other amino acids that are used very occasionally, but still, there's probably a lot more amino acids that could have been used than what actually are used. It's surely possible life could have used different amino acids. An interesting property of molecules is chirality. Many molecules are asymmetrical, and this means that they can, as it were, point in different directions. They can be left-handed or right-handed. So if you take your left hand and your right hand, even though they look basically the same, you can't sort of superimpose one on the other because they're asymmetrical. You get the same with molecules. They can be left-handed or right-handed, or to use the technical terms, levorotatory or dextrorotatory. Another way to think of this is in terms of mirror images. Just as your left hand is the mirror image of your right one, in asymmetrical molecules, the levorotatory molecule is the mirror image of the dextrorotatory one. Now, it turns out that life as we know it uses primarily levorotatory amino acids. And it's understandable why a mixture of levo and dextro amino acids were not used, because this would result in great complexity. It would require the cellular machinery to recognize both forms of molecules. Biochemical reactions tend to involve one molecule slotting into another, and obviously if both forms were used, this would be more complex. It would be more difficult to build the cellular machinery so as to recognize both forms. But why does life use levorotatory amino acids rather than the dextro amino acids? Well, we know that proteins synthesized from dextro amino acids are functional, so it seems to have been an accident of early evolution that life chose the levo ones. Similarly, the nucleotides, which are components of DNA, are constructed exclusively from dextro sugar molecules. Again, there's no particular reason why levo sugar molecules couldn't have been used. So we can see that alternative life might have had an alternative chirality. It also seems clear that the coding scheme could have been different. Here's the so-called genetic code showing the DNA codons and the amino acids they code for. A codon is a sequence of 3 DNA nucleotides. The DNA molecule uses 4 nuclear bases: adenine, thymine, guanine, and cytosine, labelled ATGC. A sequence of 3 bases codes for an amino acid, as shown here. There are also sequences which code for start and stop, and that's just to tell the cellular machinery when to start and stop transcription. Now, this code is pretty much universal. All life on Earth uses the same code, so this must have developed very early in evolution. And as far as we know, the code is at least somewhat arbitrary. Some amino acids could have been paired with different triplets of bases, and it wouldn't have made much difference. Another thing to note, of course, is that this is a triplet coding scheme. Each amino acid is coded for by 3 bases. This is the most efficient scheme if we have 4 bases and 20 amino acids. But of course, if life had used more or less amino acids or more or less bases, then perhaps a doublet or quintuplet coding scheme could have been used. It might also have used a mixed coding scheme with some amino acids coded for by doubles and others by triples, or whatever. Now, somewhat more radically, we might wonder if there could be a completely different macromolecule that plays the same role as DNA and RNA. So this is a macromolecule that's capable of information storage. So far, attempts to construct alternatives in the lab haven't been particularly successful, but maybe that's not so surprising given the complexity of the problem. But perhaps alternative life could have developed something else. Furthermore, all life uses phosphorus. Phosphorus is used in the backbone of the nucleic acids that build DNA, and it's also the primary repository for chemical energy in the form of adenosine triphosphate. The cell gets energy from adenosine triphosphate by breaking off one of these phosphate groups at the end, and so that creates inorganic phosphate and adenosine diphosphate. So phosphorus is one of the central elements in all standard life. An interesting suggestion from Paul Davies is arsenic-based life. Arsenic lies just below phosphorus on the periodic table, and the 2 elements share many properties. The main concern about arsenic is that arsenic-based compounds are much less stable than those based on phosphorus. They break down by reacting with water much more rapidly. But Davies suggests that this wouldn't be such a problem at low temperatures or in environments where there's a lot of arsenic available, and so the arsenic-based compounds could be rapidly replenished. Hydrothermal vent systems are one environment where there tends to be a great deal of arsenic available. Indeed, since phosphorus-based compounds are much more stable, organisms have to use various enzymes to catalyse the removal of phosphate groups. So Davies suggests that in simpler organisms or in prebiotic systems, the greater reactivity of arsenic may actually have been a virtue. Arsenic compounds may require less biochemical machinery to execute their functions, precisely because they are less stable. So maybe that is one area where we might find alternative life. Finally, we might wonder whether there could be acellular life that does not require the mechanisms of the cell. We noted earlier that viruses don't have cells, but viruses do require cells in order to reproduce. The same is true for other entities on the edge of life, such as prions and bacterial plasmids. But one proposal for the early Earth is that the first organisms were sort of complexes of organic molecules bound to mineral surfaces, specifically iron sulfide minerals. And then these complexes of organic molecules develop metabolic processes. And this is known as the iron-sulfur world hypothesis. Maybe we could find similar processes still occurring today. As I said earlier, we probably need something like a cell, some sort of enclosed structure, but perhaps we wouldn't need cells as we know of them. Many other proposals for alternative life have been made. The point here is just there are many options in principle for life. Life could have been different, it seems, from how it is. And so maybe there really is different life out there. So that's the case for a shadow biosphere in principle, but could it actually be the case in reality? Could there really be radically different life? A number of general arguments against the existence of a shadow biosphere have been proposed, so let's go through them. First of all, there's the argument that the emergence of life is extremely improbable. This was a fairly popular opinion among biologists up until around the 1980s or so. Ernst Mayr, for example, suggested that the emergence of life requires a whole host of very unlikely chance events. So maybe there's no life anywhere else in the entire observable universe. That seems kind of outlandish today. But that was, as I say, a fairly popular opinion. There are a lot of biologists who thought that it may well be the case that life has emerged only once in the whole universe, because there are just all kinds of extremely complex processes involved in life, even in the most basic life forms, and it seems almost miraculous that processes like metabolism and reproduction could have gotten started from inanimate matter. And so it must have been extremely improbable. It must have required all sorts of very improbable events. And obviously, if the emergence of life is extremely improbable, then the chance of it occurring more than once on Earth must be very, very low indeed. Now, these days, this isn't really such a popular argument. One of the main reasons is because of what Paul Davies calls the up-it pops objection. Basically, this objection suggests that the emergence of life can't be improbable because it arose so quickly on the Earth. Very shortly after the Earth achieved conditions suitable for life popped up, and that shows that life tends to arise very easily. So the Earth hosted water by about 4.4 billion years ago. That's the point at which the planet becomes in principle habitable. Obviously, finding fossil evidence for the very earliest life is difficult, but the earliest confirmed fossil evidence of life on Earth dates to about 3.7 billion years ago. There have recently been some reports of much older fossil evidence, but these are somewhat controversial still. But the earliest confirmed life is about 3.7 billion years old. From about 4.1 to 3.8 billion years ago was an event known as the Late Heavy Bombardment, where the Earth was hit by a disproportionately large number of asteroids. the Earth would probably have been very hostile to life during this time. So if we take about 3.8 billion years ago as the point where the planet becomes more hospitable for life, and we have confirmed fossil microbes at 3.7 billion years ago, then we can say with some confidence that life arose no more than 100 million years or so after good conditions for life obtained. 100 million years is not all that long in cosmic time. So, to quote Carl Sagan, he says, "The origin of life must be a highly probable affair. As soon as conditions permit, up it pops." Now, one problem with this argument is pointed out by Paul Davies in the afterword to the book The Drake Equation. We know that every planet has a finite habitability window, and an even smaller window in which intelligent life could thrive. So, as we noted, the Earth became habitable maybe 3.8 billion years ago. Now, due to the increasing luminosity of the Sun, all complex life will be wiped out about 800 million years into the future. Probably anything resembling intelligent life will be wiped out 600 million years into the future. So we're well over halfway through the habitability window of the planet, at least as far as multicellular life is concerned. So suppose, for the sake of argument then, that the emergence of life is extremely improbable. Suppose that on average, once the planet becomes habitable, it would require trillions and trillions of years before it becomes inhabited, because the events required for life to emerge are so improbable. Suppose also that once life has arisen, various other improbable steps are required for intelligence to arise, such as sex, multicellularity, the evolution of a central nervous system, and so on. On our own planet, life was microbial for most of its history. In this case, given the fact that intelligent organisms are here, we can expect that life would have emerged relatively quickly. So to see why this is, look at it this way. Suppose we take all the habitable planets. Now, since the emergence of life is so improbable, the majority of these will never become inhabited, due to the fact that the habitability window is finite. If it takes on average trillions of years for life to emerge, and the habitability window is only a couple of billion years, then most habitable planets will never become inhabited. So now take all the planets with life, take all the planets that are inhabited. Again, if intelligence is improbable, then the vast majority of these will never develop intelligent life. Again, because the habitability window is finite. So if it takes trillions of years on average for life to evolve intelligence, obviously almost all planets with life will fail to do this. But if you look at the planets with intelligent life, what you'll find is that the emergence of life tends to happen early. It has to happen early, because if it happened later, there wouldn't be enough time for intelligence to emerge. So if the step from life to intelligent life requires huge amounts of time, as it did on our own planet, then in general, life needs to have emerged early in order for there to be enough time for intelligence to develop. So given that we are intelligent organisms, it's no surprise that life emerged early, even if the emergence of life is very improbable. So for this reason, Davies thinks that this up it pops argument is actually just a fallacy. In order to evaluate the argument properly, we need to know how probable it is for intelligent life to emerge once life has emerged, but we don't know that. So we can't really evaluate so the fact that life emerged early doesn't tell us anything one way or another about whether or not it's probable. A second way of arguing that life is probable is to appeal to experiments which show that certain aspects of biochemistry arise easily. Most famously, there was the Miller-Urey experiment, which simulated what was believed to be the conditions on the early Earth, and then by adding a spark to stimulate lightning, this generated various amino acids, which are among the building blocks of life. More recently, a couple of months ago, it was reported that work by Marcus Rasler has found a sequence of reactions very similar to the Krebs cycle, which is a central component of metabolism, a sequence of reactions very similar to the Krebs cycle can proceed in the absence of any cells. So this may have been a precursor to life. So experiments such as this show that various building blocks of life and important processes of life occur spontaneously, relatively easily, given the right chemical conditions. Now the big problem with this, as an argument for the fact that life is probable, is that no experiment has actually generated life itself, nor do we really have any idea at the moment how to get from these building blocks of life to actual life. There are still big steps here that we don't understand. These steps may be highly improbable. So at the moment, it seems we just don't really know how probable the emergence of life is. Maybe it's extremely unlikely. Maybe it occurs easily, provided we have a good soup of molecules. We can't really say. So we can't appeal to the probability of life's emergence as an argument either for or against the shadow biosphere. Okay, a second argument against the shadow biosphere is, well, if there were such alternative life, we would have found it by now. Or at least we would have discovered some telling signs of it. Now, this is certainly true if we're talking about large multicellular organisms. I think it's pretty clear that there are no alternative large organisms out there. But we know that the vast majority of life is microbial, and our knowledge of the microbial world is extremely limited. So the suggestion is actually there's no reason why we must have come across microbes with a radically different biochemistry if they did exist. Carol Cleland, in her article Epistemological Issues in the Study of Microbial Life, suggests that the tools that we have for studying microbes may not be appropriate for identifying alternative microbial life. Cleland says that we generally use 4 methods to identify new microbes. First of all, of course, there is microscopy. Now, Cleland thinks that this is not going to be very helpful with alternative life because microscopy only reveals the gross morphology of an organism. What we're looking for with alternative life are differences at the biochemical and molecular level. But studying organisms through microscopes isn't so good at revealing that. And perhaps the best illustration of this is that for a long time we were completely blind to the differences between the bacteria and the archaea. So life as we know it is split into 3 domains. There are the eukaryotes. Eukaryotic cells are distinguished by having a nucleus and other organelles that are bound in membranes. All of the animals, plants and fungi are eukaryotes. Then there are, of course, the bacteria. And up until about the 1970s, we thought that was the extent of life. We thought that there were eukaryotes and bacteria, and that's all. But it turned out that actually what we were calling bacteria divides into 2 domains. There are the true bacteria, and then there are the archaea. The archaea are a completely different form of life. In fact, by some measures, they differ more from bacteria than they do from the eukaryotes. For example, the archaea and the eukaryotes have very similar translation and transcription mechanisms for their genes. Indeed, the standard view of the deep history of life is that the archaea are more closely related to the eukaryotes than they are to bacteria, and that's shown on this tree here. The common ancestor of the archaea and the eukaryotes is younger than the common ancestor of the archaea and bacteria. Of course, there's still some debate about whether this is right, but the point is that there are certainly very big differences between the archaea and the bacteria, but these differences were not revealed by microscopy. Now, of course, modern microscopy has all kinds of additional techniques that weren't available several decades ago, but again, Cleland is quite sceptical of their utility for finding alternative life. For example, one important technique is fluorescence in situ hybridization, where specific parts of chromosomes are made to fluoresce by hybridizing them with a fluorescent probe. And that's used to study specific DNA sequences, it's used to diagnose abnormalities or to identify the positions of genes and so on. But clearly this would be pretty useless for any kind of life that doesn't use DNA, or that uses DNA with different bases, because in that case the fluorescent probe would not hybridize with what the organism uses. And the same problem occurs for other kinds of modern staining techniques. These modern techniques were developed to apply to life with standard biochemistry. They would not necessarily reveal radically different life. For all we know then, we may have already seen shadow microbes through microscopes, but have failed to identify them as such, just as we failed to identify the differences between bacteria and archaea. A second important method is cultivation. This involves growing a whole population of copies of a single microbe in some apparatus like a petri dish. And this allows us to study the biochemistry of the microbe in great detail. Now, it's probably the case that if we'd ever cultivated and studied an alternative life microbe, we would have detected its strange features. However, our ability to cultivate microbes is still very limited. Cleland says that less than 1% of bacterial species have been cultivated. Part of this is simply that many microbes are unculturable. They just won't grow on this, given the standard techniques that are used in laboratories to cultivate microbes. It can be hard to identify the conditions that a given microbe requires to grow, and then when we do identify the right conditions, it can be very hard to replicate them in a laboratory. Obviously, it would be even harder to identify the right conditions for alternative life. We have no idea what those conditions would be at this point. We also have tools from molecular biology that can be used to extract genetic information directly from the environment. A standard technique is polymerase chain reaction. Polymerase chain reaction allows us to take a single sequence of DNA and then copy it thousands or millions of times. So here's how it works. I'll just go through this briefly. So this first stage shows a solution. In green is the target DNA that we want to replicate. In blue are various nucleotides that will be used to build the copies. And in red is what's called the DNA primer. And it's not shown here, but the solution also contains a DNA polymerase. Anyway, we heat the solution to break the bonds between the DNA strands that we want to copy, which separates them. We then cool the solution, which allows the DNA primer to attach to the DNA strands. A primer is a short sequence of nucleotides that's used to initiate DNA replication. So with these primers attached, the DNA polymerase has a starting point to begin replication, and that's what it does. So this is a very powerful technique, but the problem with polymerase chain reaction with respect to alternative life is that it requires these primers to attach to the target gene to initiate replication of it. In order to get the right primers, we need to have some prior knowledge of the genomic sequence. Now, we have primers that can be used for the DNA of all known life. These are primers for genes that encode ribosomal RNA that are shared across all three domains of life. But obviously, such primers would not recognize genes from alternative life. And more importantly, of course, this method could not possibly detect life that doesn't use DNA or RNA. And the same kind of problem occurs with other molecular biology techniques, such as metagenomic analysis, which analyzes DNA directly from the environment. If alternative life doesn't use DNA or RNA, then our standard tools would not be able to detect it. So Cleland thinks that the fact that we've so far failed to detect alternative life actually isn't such a powerful argument against the shadow biosphere. And we might support this point by noting that to this day, we are constantly discovering new phyla or even kingdoms of life. Just in the last 5 years, we've discovered numerous new phyla of archaea. If you just go onto Wikipedia, type in archaea, it has a list of some recent discoveries on the right side. So it seems there's still a lot out there to be discovered, at least within the microbial realm, and perhaps alternative life awaits. A third argument against the shadow biosphere is that any alternative life would have been driven to extinction by Darwinian competition. Maybe some different form of life could have arisen, but this would have faced competition with life as we know it, which would ultimately have led to its demise. Now, I think this argument rests on a fairly obvious mistake. There's plenty of room for different kinds of life. Archaea, bacteria, and eukaryotes are all very different, but have coexisted for billions of years. Most forms of life have very little influence one way or another on your chances of survival and reproduction. The vast majority of bacteria, for example, are no threat to you. They place no selective pressure on you. And of course, plenty of bacteria are actually useful to you. We live symbiotically with them. The same is true of the relations between microbes. Microbial communities are very diverse, so we tend to find lots of different microbes living symbiotically. Bacteria, archaea, and eukaryotes can all coexist comfortably. Certainly they can coexist without driving each other to extinction. In any case, even if we grant the premise that life as we know it would tend to outcompete alternative life, the alternative life could still exist outside the limits of standard life, and then there would be no such competition. So maybe alternative life could exist in environments that are too hot, too dry, too acidic for standard biochemistry. The upper limit for tolerance, for temperature tolerance of standard life appears to be about 130 degrees centigrade. So perhaps alternative life could thrive at much hotter temperatures. The fourth argument is that the emergence of life prevents the emergence of any other alternative life. This idea was expressed rather nicely by Charles Darwin. When he imagined the conditions for the origin of life, he said, and I quote, "If we could conceive in some warm little pond, with all sorts of ammonia and phosphoric salts, light, heat, electricity, etc. present, that a protein compound was chemically formed, ready to undergo still more complex changes, at the present day, such matter would instantly be devoured or absorbed, which would not have been the case before living creatures were formed." So the point is, if you imagine a pond containing all the right chemical constituents that could build life, life might be able to arise out of these constituents by some simple process. But if life already exists, those chemical constituents will simply be eaten up. They won't have the chance to come together to form new life. So the argument is that once life has arisen, new forms of life are prevented from arising, because all of the useful molecules are just used up. Now, I think that this does present quite a serious problem for how alternative life could have arisen. How could a second genesis have occurred? But there are in fact a few options here. First of all, perhaps life emerged at around the same time on different areas of the planet. It will take a while for life to spread, especially if it emerges in one specialised environment. So we might suppose, for example, that life arises around a deep hydrothermal vent, so the life there was adapted for the extremely high pressures and temperatures there. It may have taken quite a long time to spread into other environments. Then you can suppose that on the other side of the planet, maybe in some shallow clay riverbed, a wholly different environment, a completely different kind of life emerges. So now we have two different forms of life evolving separately. If the emergence of life is a relatively simple matter, and so is highly probable, it would not be surprising if something like this occurred. Now, as we discussed, we don't really know whether or not it's highly probable, but if it is, then this kind of scenario would be plausible. A similar scenario concerns the late heavy bombardment, the period of massive asteroid impact that I mentioned earlier. Suppose that life emerged early in Earth's history and spread around the planet. Then suppose that impacts during the late heavy bombardment sterilised the surface, wiping out almost all life. Only extremophile microbes survive, maybe in the deep subsurface. We know today that life extends about 5 to 10 kilometres below the surface of the Earth. In fact, subsurface microbes account for about half of the Earth's biomass. So anyway, with the surface sterilised, once conditions became habitable again, maybe life could have arisen a second time. Another option is that alternative life could have developed on Mars or Venus, or maybe even somewhere else, and then have been transported to Earth by meteorite. This so-called panspermia hypothesis was actually taken quite seriously, even independently of any considerations concerning a shadow biosphere. So we know that asteroid impacts can cause chunks of rock to be passed from planet to planet. We've discovered Martian meteorites right here on Earth. We also know that some extremophile microbes could in principle survive the journey through space if they were embedded in a meteoroid. Some bacteria can be extremely resilient to extreme conditions by forming endospores and entering a kind of dormant state. This dormant state can last for thousands or millions of years. The biologist Russell Vreeland managed to revive bacterial spores that had been dormant in salt crystals for 250 million years. Raul Cano and Monica Barucchi revived bacterial spores from a bee trapped in 25 to 40 million year old amber. So that long journey through space could be survived by a bacteria that enters a state of, or a microbe that enters a state of dormancy. The main problem, of course, with this idea is the initial impact. But there are various tests which have suggested that some microbes could survive the sudden shock of the initial impact and then the shock of the landing at the end. Obviously, most microbes would probably be killed by this, but you have to remember that in any meteorite there could be millions or billions of microbes. So it would only take a few of them to survive to populate a planet. So it is plausible that life could have emerged on Mars or Venus and then been transported to Earth. Earth might then contain two types of life. Finally, of course, it may be that there actually was room for radically different forms of life to evolve in the same places at the same times. Although it's true today that any useful molecules will be quickly used up, early life on Earth may not have been especially efficient at consuming resources. Evolution seems to have been fairly slow in the ancient past. Perhaps there was a long time during which life was very simple and it only used very specific resources, so that other resources were available for the emergence of other kinds of life. Early life was not nearly as diverse as life is today, so maybe there actually would have been useful molecules that were not just used up. So arguably then, it looks like it is in fact possible for there to be a shadow biosphere. The question is, how would we go about finding it? How might we go about getting empirical confirmation of a shadow biosphere, if indeed there is one? So I'll just mention a couple of possibilities that have been raised. So one possibility is to focus on currently unexplained phenomena. Carol Cleland cites the example of desert varnish. Desert varnish is a hard, dark coating found on rock surfaces in very arid environments. It's extremely thin. It's around 1 micrometer thick, and it's separated into chemical and mineralogical layers. In this respect, it resembles the microbial mats or stromatolites that were among the primitive forms of life on the Earth. Now, there's currently no consensus about how exactly desert varnish is formed. So Cleland suggests there's a possibility that this may be a trace of alternative life. Most intriguingly, desert varnish has been found to contain very high concentrations of manganese and iron, while the underlying rocks have very low concentrations of these elements. Manganese and iron are commonly associated with microbes as a byproduct of metabolism. So there are some indications that desert varnish could have an organic origin, although it doesn't seem to be produced by any microbe that we currently know of. Another suggestion, quite controversial, are nanobes. So nanobes are small structures first found in rocks and some sediments by Philippa Hewins. They're only 1/10 the size of the smallest known bacteria. The smallest nanobes are only 20 nanometers in diameter. They're too small to contain the appropriate biochemistry and biochemical processes needed to sustain life as we know it, because the minimal size of a cell for life as we know it is said to be around 200 nanometers. So if they are living things, then they must be an extremely unconventional form of life. What's interesting about nanobes is that they are non-crystalline. They're composed primarily of carbon, oxygen and nitrogen, three elements associated with life, and they appear to be capable of spontaneously growing and spreading on different surfaces, forming colonies. Ewens claimed that on close analysis it's possible to identify an outer layer or membrane that may be analogous to a cell wall, which surrounds an electron-dense region that may be analogous to the cytoplasm. Again, as I say, that is very controversial. This is not widely accepted in biology, but if these are living things, then this may be an alternative form of life. Related to nanobes is the idea of nanobacteria, which are proposed living organisms that are morphologically similar to bacteria but much smaller. These kinds of structures have been confirmed and they do appear to replicate. Intriguingly, these structures were discovered in pathological calcifications like kidney stones and atherosclerosis, leading some scientists to suggest that they might be implicated in these diseases. The general consensus these days is that nanobacteria are not living, but arise due to the crystallization of minerals. But, who knows, maybe further research will change those things. So, nanobes and nanobacteria might be two possible possibilities. Another option for finding alternative life is to look for life outside of what we currently think the limits of the biospace are. We look for life where it's too hot, too cold, too acidic or alkaline, or too saline and so on, for standard life to exist. So I mentioned that we think the upper temperature limit for standard biochemistry is around 130 degrees centigrade. There are some hyperthermophile archaea that have been found up to 122 degrees centigrade, but nothing beyond that. So if we found microbial life in a deep ocean volcanic vent at 160 degrees centigrade, that may well indicate a new life form. Finally, of course, we could try to culture life using biological filters. This is perhaps rather less likely to be successful. As I noted earlier, we struggle to culture even familiar microbes. But if we could find some means to interrupt the biochemical machinery of standard life, any metabolism that continues may be alien. An obvious option is to use chirality. We noted earlier that standard life uses levorotatory amino acids and dextrorotatory sugars. We might try using a solution of only dextrorotatory amino acids to try to culture life. If that was successful, it would strongly indicate an alien life form. So that is the shadow biosphere. It's an interesting idea. Who knows whether or not it's true, but that's something to think about. I hope you found that interesting. Goodbye.